showcasing the really innovative and important research and scholarly work being done across our community. We're delighted that our first presentation this semester Hartert. Dr. Hartert is an associate professor here in the Department of Biology, completed his BA at Carleton College in 2013, PhD in Nebraska Medical in 2000. Following this, I completed a postdoctoral fellowship at Mayo Clinic in Rochester, Minnesota. Through his research, Dr. Hartert aims to identify genetic alterations that drive lymphoma and inform treatment decisions, validate mechanisms of biology, translate results in action. To these ends, he is specifically interested in the assignment of personalized lymphoma therapeutics based on genetically characterized tumor biology profile. His studies have primarily focused on understanding the most common type of lymphoma, diffuse large B-cell lymphoma, or LBCL, which accounts for 30,000 patients each year. As some of you know, and I will confess that before preparing the talk, lymphomas and leukemias present a unique challenge because they do not form a single solid tumor in a concentrated area. Equally challenging, DLBCL tumors can evolve from distinct genetic origins that result in heterogeneous biological profiles. Using genomics, Dr. Hartert's research helps us define the genetic landscapes of leukemia and assign a targeted therapy best addresses that biological profile. I'll also add that as a scholar whose work is rather far from biology, I'm trained as a sociologist, I especially appreciate Dr. Hartert's research when he makes it accessible very often. For example, over the weekend, I listened to an episode of this podcast, also titled Cell Tolls. I was amazed at how much even I know. I'm really ready to learn more. Please join me in.
Thank you, Dr. H. Been waiting to say that too. So, yeah, I'm gonna use this title as long as I can get away with it until some copyright comes after it. I thought of it in 2018, and I was like, "This is it. This is my big ticket." And yeah, I mean, I'm still pretty happy with it. Let's be honest. All right, so exciting stuff. Good news too. I know I actually gave away too much of my intro here, but yeah, the only thing I think I can contribute is that I'm from Colorado and it's super pretty. Hooray! Learned how to hike, high altitude, good times. But yeah, went to Carleton, UNMC, Mayo Clinic, and now we're here to teach. We're doing a good job. It's fun. But alongside everything, still got the research side of things that I always wanted to kind of pursue. Now, when your immune system, that's like kind of where we're going to see why this cancer is such an issue. Because all your good guys in your immune system, they can always go bad. So all of us have these little things, B cells right here. Should they be told what to kill? Something specific. Y'all have heard of immune memory before, right? They will form these things. Giant antibodies, these little Y-shaped things, they can hook onto viruses, bacteria, parasites, tumor cells in some cases. But transition from here to here is fraught with all sorts of dangers. The night is, what is it in Game of Thrones? The night is dark and full of terrors. And man, a germinal center is nothing but friendly. When you begin your journey as a B cell, you enter in your lymph nodes or your spleen these nasty sort of like engines of cells. Should you be lucky enough to even make it to this transition right here, you're allowed to expand. What's the first thing that tumors do? Right, expand. See, we're already kind of messing with things that kind of dark forces for ourselves. What happens next doesn't happen anywhere else in the body. Let's see how sort of a cycle is forming right here. There's a few things in here. It's going to take your DNA to make that antibody thing that we just saw. It's going to take the genes that make that and mutate them, translocate them, switch and swap them until eventually meet whatever the antigen is that you want to have memory against. There will be one winner in there, and that cell will eventually become the adult called a plasma cell, and it will shed memory antibodies, and the antibodies overwhelm pathogens. That's why it takes about 3 to 7 days to stop being sick from some of this stuff, is because this process is violently happening in that moment. A kind of example I always put for this is, who's ever heard of the shark that it has offspring in the womb, they all fight, and one comes out. Yeah, it's kind of neat when you hear about that. That's kind of what I always was like, that's kind of what happens here. So it makes sense that this is the 7th most diagnosed cancer when it's the only place in your body that genes are mutated for that reason, to make them better. But that's another reason why the antibodies at the end, not purely unique, right? No human had ever had COVID on the planet. We eventually, half of us may be right here. Okay. Whew. A lot of fun figures, but mostly you can focus on the text at the top. There's 3 aims for this where we're at with this disease. Number 1, how I kind of integrate things. Number 1, we have tons of data, patients, genes, microenvironment signatures, everything. We can try and track and see not only who dies and lives, but who would respond to certain drugs and who doesn't. Not just gene mutations either, it's who's up, who's down, it's who's kind of in the wrong place sometimes. So let's say that we find targets in the data. We go into the cells and we'd actually say, is this gene actually offensive in the cell? Is it actually driving a bad deal? That's where we start to do like Western blots, where we actually see there's actual protein growing for cells, for example, or reducing if we treat it with a certain drug. Then the third one, most people's favorite, translate. So I choose unique because I was mentored by oncology. So there are plenty of labs. There are plenty of labs and here, but our goal was always take the next step, capitalize, don't wait for somebody else to do it. Find a therapy that, based on what you found, this is going to work for this type of patient. That's where the goal with precision medicine comes in, is that every patient's tumor is different. That's a strange thing to hear sometimes, that no matter what, different genomics, circumstances, environments, we have the drugs to attack it, why not go for it? So, I've already seen this a couple times, but this is what you would call the hallmarks of cancer. In today's talk, the 2 basic ones that pretty much any really, really bad cancer is ever going to need, you need more cells. You need to massively increase that amount of tumor. And with 1 cell becoming 2 in the normal process of mitosis, this is out of control here. But that's not good enough. You can't just have a lot. They can't die. Those 2 are the main 2 for any cancer biology right there. Today's kind of fresh take that we're going to be looking at, that these things have to avoid being hunted by the predators that are still trying to get them in your immune system. It's where ecology actually strangely makes its appearance in tumor biology. Very cool. So, getting along here, a slight review to say the least. Those of you in genetics are excited to see it from the slide, I imagine. Most of the time, y'all got normal genes right here. Comes out, nice little functional whatever it is. Should you change the DNA, though, in this case right here, and make a little fizzle, oftentimes you're going to get something that doesn't work or something that's totally defunct that we work with. So losing genes, typically when you mutate stuff, you're going to lose them. There's going to be the case where you mutate things that they sort of gain a function as well. Most of what we'll look at today is lost. So, eventually, you're going to take a normal cell. If it has too many of these mutant states, eventually it gets out of control. The wires cross. It's no longer basically being run by, the prison is no longer being run by the guards. Things are out of control. The cell behaves sort of like a wild animal. It just sees what genes it has, goes. And a cell that just goes is going to make more progeny and offspring, and they can take over in a lot of cases. So, onto the disease itself. Big old mouthful. There's only one, people always are like, is it Hodgkin's or non-Hodgkin's? Most lymphomas, all of them except Hodgkin's or non-Hodgkin's. This is the biggest one. You can get anywhere between, based on the estimates, you can get anywhere from like 30 to 40 to 50,000 cases of this. Like I showed you, that's why it's such a big deal with that immune like recycling. These things are constantly being refined, but taking on damage in ways that you would want. The staging is a little different than most other cancer stages. Most other cancer stages in stage 4, it's spreading everywhere. Lungs, brain, all that. Can you do surgery on your white blood cells? You just like, you're like, we're gonna cut this tumor out. No. It's already everywhere. That's the big problem. Even though we still have stages, notice it's usually just going to be about the location and number of nodes that will form. But these things are circulating everywhere throughout your circulatory, and they're native white blood cell highways. Not ideal to say the least. So we had to turn, and I say we as though I was there like 50 years ago, we had to turn to chemistry and big stuff to systemic things to burn this out. To be honest, it turned out pretty well. Did I try the light on this? Okay. Contrast. All right. So, looking where we're at right now, pretty good. You always get a few questions from people. It's like, where's the cure for cancer? They're keeping it from us. A lot of time, chemo can do fine. You're looking at right here with R-CHOP. These are 4 chemo drugs right here, followed by an immune therapy, rituximab, that kills a thing on B cells called CD20. Nice and targeted, nice little synthetic antibody. As you can tell with the survival curve, about 2 years to 24 months right here. 78% of people still alive, pretty good. 5 years, see how that levels out almost with like 5, 60 probably. Put it around right there. And as you can tell, about 50% of people essentially have what we call extremely long-term remission, which is also code for the word in a lot of cases. Wonderful. Now, unfortunately, if you end up in anywhere in this zone down here, we're going to see what happens. The good news is, should the R-CHOP not work, sequencing gave us a view of the entire set of pathways that are driving these bad things. We looked at all these genes, we found all kinds of ones that are offenders. These are the ones that we call oncogenes, where they're doing a bad thing. They're driving the issue. That means that we can drug here, 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 right there too. We can send all kinds of treatments at them. Doesn't mean we knew why they worked each time, that all were effective, to say the least. Plenty not. So remember, I showed you that group of patients, that 50% that doesn't sustain the long-term curve or long-term cure. Should you end up in the next refractory or relapse sets, that's what you're looking at. Right in a year, you've already lost 80% of people. At about 4 years, not much change right there. It's a steep drop. So when this disease happens, it happens bad. Sort of a coin flip, to say the least. Not one any patient wants to take. This is where we were at in 2017. Now, what happens next? It is my field, but a pretty big deal. All right, sci-fi wise, this is not a normal drug. This is taking your own native immune cells, taking them out into a lab, giving them gene cocktails, essentially to supercharge them and genetically modify them and put them back. They hunt the tumor. We give them synthetic genes that don't exist in nature. They go through and they clear things out. Clear things out so badly in some cases, go into a sort of delirium because the immune system is so powerful. So we've started to, I want to say, almost master this. This has been phenomenal, to say the least. That's the new survival curve. Remember last time we left it a year, we were about 20% alive? This is only the beginning. 20% to 60% jump. People get published in the top journals for making a 5% jump. So this was a totally new step. This wasn't drugs, this wasn't chemo, this was refitting the immune system with something it had never seen ever. The data's still rolling in. It keeps getting better. The competition in the market is so aggressive. Now, despite the new hope here, despite the new hope, still a long road. Looking at here on the other way, understand. The next thing that we've got working for us not only is this new massive therapy, but it's the fact that we can sequence so cheap now. We're starting to sort of cluster tumors in patients into distinct genetic family groups. That's technically what is happening in these heat maps up here. These are genes in the rows, those are patients in the vertical. You start to see how they kind of cluster together. Families sort of form based on what alterations exist. Beautiful heat map by both groups. shows where we're at. We're starting to see that some tumors are highly related while others behave sustainable. So, this is the beginning of that personalized therapy step. This began a little before the advent of CAR T. They sort of met right in 2018. But the whole beauty of the idea is that you're going to be able to take a patient, take a look at their tumor, classify them, take the exact right drug. They've even gone as far as adding drug targets for each group. So for example, this STE-driven group would respond to PI3K inhibitors, bad gene, and JAK inhibitors, another bad gene. We're finding that this information is guiding. Yeah, that's sort of where I came in at Mayo. That was my project, to sort of see who's not making it, what families are bad. Now, I took this and I wanted to go a different direction and sort of be the fast fish to uncover this beforehand. But I showed you how good CAR Ts were, right? That means that the ones that aren't doing well still need something. And it also means you can help it. You wouldn't want to take the CAR T, by the way. It is about $500,000. Number two, like I said, the side effects can kill you. Probably going to take that 60% shot over the 80% shot, right? Given that, though, we can find these patients and assign something different, something a little more targeted in that case to them. So here's the original napkin of who fails. Put this together after, I think, the 2020 American Society of Hematology meeting. As you can tell, those of you that had me in class, my writing is sort of like a little Da Vinci code, but we can read it. You guys are good at it now. There were two things circled in the best. The immune system had to already be dead in a patient. That's what I mean by cold. And that's also essentially what we find out 2 years later. It's the same phenotype of what we call a deleted tumor microenvironment, TME. All these other things could lead to that metabolism, MYC gene, pre-mutations, CDKN2A. That's who I'm after. I want to know people. I want to know patients that they arrive with the relapsed bad tumor. They shouldn't have to go through CAR T. Something alternative. All right, this is a lot cleaner. Sorry, but I wanted to use my napkin. But essentially, whatever roads that lead to tumor has basically overwhelmed the immune system, and it's like a desert. You don't want to send in CAR Ts there. They're just going to be helpless. They need to actually command and tell the immune system to do something. Without any soldiers, the generals are useless. So, as we go through here, my friendly data sources, including this gold mine that I found about a few weeks ago. More on that maybe in a year. So, let's meet our first loss. This gene right here, P53, TP53, it is one of your best savior genes. One of your cells starts to go bad, it saves you. Close the gene up. Now, if I destroy this, though, that's what you're looking at below. You see the survival curve, you have P53 versus you don't. You also see for the CAR T patients right here, you have it, you're red, you're failing. You don't have it. Yeah. Well, not forgetting on this side. If you have P53 mutation, like it says there, the coin sort of flips from 60% good to 60% bad. That's a 20-point shift. Not ideal to say the least. So, first thing we did was look, if I take P53 loss and I say, what's that associated with? One gene showed up incredibly high. My wife likes to call it BIRD4, but that's unprofessional. BRD4. Oncogene is sort of a partner that shows up after this loss. So on one hand, we have a bunch of pathways over here that when you have P53 loss, you lose a ton of stuff. Gain others. So the green right there, you see a lot of gene gains. Fair enough. You get a bunch of metabolism stuff, a lot of Krebs cycle and glycolysis, our favorites. You lose all of your cell death stuff. What that means is the cell can't die anymore. Bad time. In pink, immune regulation. Strangely enough, you lose P53. Remember all that big deal about the CAR Ts? You're going to lose all this T cell stuff right here too. It's an association. And that's what's worth investigating over here. Given that, first thing that we saw was genes that were high to the right. If you had P53 mutation, BRD4 very far to the right, high up on the axis. If a gene is associated, what I call EFS24 failure, Mayo Clinic metric, or if fail within the first 24 months. Should you fail within the first 24 months, the rest of the months interval, maybe 24 months, you enter a phenomenal population. As you can tell, BRD4 is high, fails, high problem. Even alone, BRD4 is not ideal, significantly, to have as a high gene. So this partnership was previously unknown. Another study comes out. This is one of my favorite papers that has ever happened. A guy took, well, a group took CAR T cells, knocked a gene out each time, and saw what genes matter the most for these CAR Ts to work. A few death receptors that we knew about showed up. This one right here, TNFSRF10B. Sorry, call it TNFSRS eventually. You lose that gene, you're toast for CAR T. It's not going to work. They can't even find it. His paper went on to go off and say, we can actually renew this gene, we can do all this stuff to bring it back dead a little bit, but kind of end it. So, we did sort of a triple threat then. BRD4 is high. P53 is mutated. Who do we lose, and who is the most important in size for that CAR T score to work? And also, who is a oncogene versus a tumor suppressor? Tumor suppressor is a good guy. You don't want to lose that. PKN1A is key, but classic tumor suppressor. That's bad. It stops mitosis for you. Not ideal. But then among all these other genes, pops out nervous, NFS. It's a full loss. IPRD4, down for this gene, alter, going down to. That is not ideal. We're sort of triangulating on some of these why questions. Why did this not work? Here we go. This is fun. We got a busy slide. You guys love this though. Okay. Next, when you're P53 altered, how does your immune system actually behave? We can take raw gene expression values and interpret and sort of deconvolute that, is a cool way to say it, and interpret the numbers of the other immune cells. First thing we did, we just went kind of boring. We're just like, all right, if you have P53 mutant in red, here's all these genes for good pathways for your immune system. See all those reds go down like that? Terrible. Same thing over here. You lost that derba gene, TNFSR10B. That was not ideal either way. These are all a bunch of genes just that aren't immune genes. Single genes right here, sort of just validating that. Here's where things get fun. Low TNFS10B, P53 altered. That means that these are getting lost right here. Double loss. If we examine these, CAR T cells need one thing, and that is good T cells running around in that bad pile. All your killer T cells, your innate gamma delta T cells, strangely neutrophils, they were a surprise. Maybe more on that some years later. Data's data. Similarly, you're also losing some good macrophages and zeros. Essentially, all the good guys are already not there. Not waiting. So if you infuse CAR T and you have mega losses of these, the CAR T will explode around and die. Patient dies. So, given like kind of where we're at, we've triangulated these gene signals. We've also now seen the associations with the raw immune cells themselves, because we've gone beyond the tumor. This is just your normal cells, and we can infer their location and their quantity and their power with the same data genomics. We would go eventually further as we took a look and we're saying, how did BRD4 also kind of ruin things as well? It is a direct bad correlate essentially with NSF10B, but mainly that key tumor suppressor CD10A1A. Again, network, and trust me, most projects don't have this big of a network. It's a good and bad thing. Makes for very one thing. But so long as as this is high, notice that red, the others start to go in a sort of opposite direction, right? The purple and the green are our two genes that are friendly for us. Notice where all the P53 mutations are piling up back here, right? All where this is high, these are low. As you have working P53 in white, a lot happens. Gene partners that it had, especially with the very critical network, CDKN2A, massive connections. So what this figure over here was saying was like, who in the tumors with low CDKN1A, that's bad. Who are the top associated genes? There it is, just waiting all the way at the top. Put this in perspective. That is 30 zeros behind the significance of what you'd expect. You know how p-value is 0.05? You like 30 zeros before 5. We're talking about. So now we're in trouble though. Because we can drug BRD4, right? It's a bad guy. We can send a drug and kill it. Can we bring back top genes, as we talk about in genetics, right? Somebody asked this in class, didn't they? And I kind of was like, that's a story for another time. It's this story. This is new. Most drugs go in and destroy a gene. They destroy the protein. Prenopop, something 53, being a stop gene. This drug right here does something different. It doesn't kill the gene. Most P53s mutate in the same spot. It actually refolds it back to its native form. It makes it work again. It brings it all back. This won't work if mutated like most are. We were excited to give this a shot. We paired this different gene. Edas-Nutlin, basically, kind of a little name. What it does is kills one of the killers of P53, MDM2. So if we destroy this, and bring these levels back up, bring these levels back up. This guy, want that surface there. That's what the CAR T cell needs. Like we showed with the Prenopop, that good guy, it's going to bring things back. We're on to that second step, validate and translate. We found these targets. We had to see that it worked. Taking both of those drugs, took them up against 6 of our tumor cell lines, which we keep and grow in the lab. As you can tell, when we started to add more inhibitor in each case, they went down, some more sharply than others. Specifically, all of the P53 mutant ones responded exceedingly well. Here's the best part. Check this out. There's the 2 drugs down there. These are noted. They were applied in each treatment. So, in DHL6, one of those ones up there, there's your start for TNF-DASV. The actin is just a control. That's supposed to stay the same time. But we had nothing. We get the native amount, not great. We add just a Prenopop, kind of cooking. Yeah, Edas-Nutlin. Things are going a little better suddenly. We had both. Comes back. Same thing happened in U2932. Back to the dead. Pretty neat to say the least. I can't say that I expected that to work so well. There's plenty of times that Western blot does not work. These take like 2 days, and sometimes you just stare at the screen and it's like, wish I had the 48 hours back. This is a good one. Next, I promised you that I had to give you a translatable therapy, right? I've got it. So, a second one, a little more aggressive this time than the first one. 2 genes here. Let's say that we wanted to kick this out, this BRD4, we set it up against a drug that kills it, ARV-825. This right here, CDK4/6, and Mesiclib, you've actually probably seen that on commercial advertisements lately. This helps the pathway that was connected with BRD4. Remember the CDKN1A loss. This 4 and 6 CDK rises in massive numbers with that loss. Drug puts it back down. Right here, you can see great single cell responses for each one. Excellent. But here's where things started to get exciting. The clinicians get excited. The red regions are areas where, instead of just what you'd expect, if you killed 5% with one, 10% with the other, you put them together, it's like 80. That's what you call synergy. Drugs working together that wouldn't expect that good together if we're an HD red region. Equally, microscope sort of confirmed what we were seeing. The MSO is just treatment that's nothing, single drug, same amount of combo. Big old desert, tons of blown up. Pretty neat to say the least. Are good. Can't zoom in, I can't just sit here and zoom. Now, what ARV was, that drug that we killed BRD4 with? I chose something a little aggressive. It's called a PROTAC. That's the little name for it. Remember how I told you most drugs, so like imimesiclib behaves like a normal drug, goes and finds CDK4 and 6, plugs it in, kills it, that's it. What the PROTAC actually does is it takes BRD4 right here. PROTAC itself is a linker. A linker finds your cell's natural garbage recycling center, ligase. That, you know how you make genes and you gotta get rid of them eventually, right? This puts those two things together. Make it complex. Suddenly now the E3 ligase is like, hey, I'm gonna kill this thing right here. In this case, this is that bad BRD4 that we just saw. No more BRD4. But there's a catch, unfortunately. Makes this a little tough to translate. When we would put this in the mice or humans, do you notice how the linker just recycles and does it again? Pretty good. Sadly, in vivo right now, don't need that much. They don't know how to control it yet. Now, the other problem here too. Remember how BRD4 is a growth gene, a survival gene, a bad guy for the tumor. The B cells in our tumor are pretty related to the T cells that we need to hunt them. BRD4 is also in the T cells we were hoping to show up. So that means that this treatment is only worth using when the patient has no T cells already. We just got to kill that tumor. It is a little niche. Precision will get that way. It's a good look at the future of how we can eventually make cancer chronic someday, potentially. Something soon where you just go therapy to therapy to therapy. 3 years at a time, 4 years at a time. Rotates. Can't use the cure word yet, but it's better than nothing. All right, brief little journey. We're taking one more look at the napkin. I still have like 10 minutes or so. We're good. Now, we're going to flip the script. Before, losing P53 was like you're losing one of the good guys. Let's look at what it looks like to have a nasty guy. Nick. This is a gene that drives all kinds of bad stuff. If you have a lot of it, you're in deep trouble right here. You mutate it, or you get too much of it. Too away. Bad time. That red line is not somewhere you want to be. Similar to what we saw with P53 with CAR T patients. About 65% of them are going to fail. They have this high version versus our larger now updated population. It's about at least 50. Equally, same sort of idea kept happening, like why this was associated with CAR T issues. Same thing wearing green, driving proliferation, you got some nutrient stuff, cell death stuff that's going up and down for bad reasons. And then the pink again. You have high NIC, you're losing all those T cell good genes. The story's pretty similar at this point, except we gained this thing. This is a totally different wing. That's the other strange thing. This is a totally different genetic family than the tumor we just saw before. That's the challenge of precision, is that we're still in the same disease and we're only on one of two little genetic families. So here's something though that represents a weakness. You know how DNA makes some RNA. The RNA has to go out and become a protein, right? Get that RNA out. Don't worry, we don't have to do this quite in genetics yet, don't worry. You have to get out of the nucleus. The nucleus is super protective. The little XPO1 gene would later help us identify a way to destroy it. See how it's helping it? We can't drug it, but we will be able to drug that in these slides. All right, here we go. Next. Got new fresh data for this project where I had somebody named Schaller. She put together gene sets for who responded to CAR T cells and who didn't. Now we're working ahead from the last project. The genes that are low on this axis down here are lost in the people that died despite taking the CAR T. Now look our X and see what's low. When you have high MYC, goes low over here. Then CAR-T score again, how important are these genes for the CAR-Ts to work? How associated are they with failure? So that red darkness means they're associated with failure. And they're lost. Here we go. Take a look at some of these fun ones right here. That is a key T-cell modulator right there. You need IL-7. That is a key T-cell signaler right there. That is a key stopper of a stopper for T-cells, so you need that as well. For NK cells, good macrophages, good immune response. T-cell co-stimulator. So again, back to the same thing. Why all those roads the original napkin led to immune system dying? This gene is part of that association, but how was it deriving it potentially? Again, similar shot here where if you had high MYC, you were losing some of these critical single genes. You saw that ILR7, you saw CD58, you saw CD28. Every time you have that MYC, these patients had that bad look. Similar again, have high MYC, driving this way. Failure association, we took the data and said not just genes, give me cells. Here we are again, down here in the dumps, losing our killer T cells again. Again, the neutrophils curate once more. Like I said, didn't expect that. Potentially someday we'll have a good story on that. Losing these again means nobody taking CAR-T. Should be taking them. You have no CD8 T cells to tell who to kill. Okay. And here's where things got a little more interesting. I know I got 4 curves going on here, and that's a little, I don't know, a little break for me. But in black is most of our patients. Normal MYC and CD8 cells were present. Take a look at the teal and the red. High MYC would be bad, right, in teal. We actually see they do okay, because in this case, they at least have good T cells running around, helping, patrolling. It's fine. Should you lose those T cells in red, because the people had the high MYC gene, the T cells were also gone. That's where things have to drop, right? Within a year, you lost 40% of those people right there. Now, here's the other problem with precision medicine. Groups will get smaller as you whittle away and you filter away more. We're only working with 19 here versus 30 in this case, but at the same time, significance for just this versus everybody was still significant and significant versus this for any group, significant against the others. So, combination of the two, that was the key. Here's the latest development, where this is all we're going to. You don't have to see Bowenstein's really good pictures right here, but what you can do is the nature of what's changing in the field is that we've been sequencing tumors for DNA and RNA, right? Totally fair. It's the cells around the tumor, though, non-cancerous ones, matter, not just the immune cells too. But what she's defining is microenvironment. What this is, like I said, this is essentially ecology. There are different animals running around. There are different environments that are better for certain animals. Tumor being one. They think of it as an invasive species. So she coined the word ecotype. Now, strictly speaking, that the worst ecotype is the one that we've been seeing. Depleted CD8 cells. No hunters. The invasive population. There it is, right there. CD8 T cell response for the drug. So that's always a distinct thing that is sort of troubling at this stage for everybody, is that, and for our field, which was everything we've done to this point has been classifying the tumors themselves, when in fact, probably the more important thing has always been the cells around the tumor, right? How they inform them. Sort of a good little endocrinology shout out, right, Dr. Cohen? So, when you lose MYC, here's everything you're losing for all those good CD8 things. And actually, not only when you're losing MYC, but when you have low CD8 counts, you can't form blood cells, you can't activate them, you can't differentiate them. You can't get the T cells helping. All of these little bars are saying how significant of a loss that was for that group that we saw. Then my favorite figure, and even though I had to whittle it down to 19, we took Dr. Steve's cell states and applied them to all 400 of our patient tumors. And we get to make some of the best XY plots you've ever seen. They don't tell us a ton, but I did have to show these. So MYC versus CDA gene expression just in the tumor microenvironment. It's a negative association, even though I do have log axes so that it kind of didn't like squish. And if you take a look at just the 19 patients that we had high MYC and low CD8s, see that those large bubbles as well to note that those were failures. For 24 months, they're all over here low, basically. Same thing happened for each one of the genes that we wanted to test MYC versus. This is another one of those BCD8 genes. Again, not an ideal look for where you want to do risk patients. Fun. All right, who's ready to translate? Who's ready to kill this thing? So, MYC itself is one of the quote-unquote undruggable genes. Doesn't have a pocket to fit, but we did two other things. We're going to go after one of its targets, CDK9. We can kill that pretty well. Even though CDK drugs are in their infancy, they're really powerful. Now, I told you about that little thing that helped MYC out of the nucleus and become a true gene, XPO1. We can't drug MYC. We can drug its partners after. We can definitely drug its partners before. Cornering it. We proposed that this would work. We were happy, and I say the word mixed. None of these like existed. They all eventually died. I say mixed in that there was a sort of heterogeneous response, and heterogeneous meaning different in some cases. But in any case, when they had a MYC alteration, they responded extremely. Yeah, going down. Yeah, and specifically some a little more than others, to say the least. So, once again, the translation finishes nicely. We got even better microscope pictures this round. Sorry if we're here again, but yeah, results are good, right? And these figures only come in one format, so that's okay. But again, when you put these two together, MYC is completely cornered. They were addicted to it. Now they're all dead, right there. Synergy ratings, you need about a 5 to be considered synergistic. 5.04 barely got it. But then over here, in one of the most vicious tumor cells that I have, like 33, massive. And then again, the beautiful look at the controls, single drug, but then beautiful. So I suppose if I wasn't so much of an animal person, I would plug these into mice, but I can't handle that. My heart can't. We're not going to go that far. So here we are. And the cool thing is that we've been able to sort of publish each of those two main steps. One frontier that I still am working on, one last one, and that project is still ongoing. Metabolism and driving. We figured that out. Figured this out. Okay, next. Now, here's the other cool thing. A couple things that I learned from the most recent ASH meeting I was at in December. The villains of all of this turns out are those ecotypes in the environment. The macrophages can go bad and turn pro-tumor and serve the tumor instead. Tumor will stick signals out them and sort of convert them. Remember that time in Jurassic World when the T-Rex told the raptors kill them instead? A little bit like that. Here's another thing that me and my graduate student Adam are doing. Instead of drugging these things, since we're starting to figure out, we've started to like corner the market on precision and who's not going to do well with CAR-T. There's a new emergence happening where we're going to send in certain drugs to drive the CD8 cells into a frenzy, get them rolling a lot harder. This will be a different sort of thing for us because I'm not used to drugging a cell then pitting a cell against the tumor cells. That will be exciting. And one last strange thing that we saw in the meeting. When they would do some of these really advanced single cell RNA things, where you're getting every cell, getting all the genes for single cell, we don't have one of those genes here, but we have their data. People that responded had a very odd, about 5 to 10% B cells that weren't tumor. People die didn't have this. The crazy thing is that in these tumors, people that survive have a few non-cancerous good guy B cells fighting the good fight, like guerrilla fighter, essentially. Like, come alive when they're given the right shot. Kind of nuts. There's more where this is coming from. I got to do a podcast episode soon. I'll get it later. All right, here we go. We did all our ASH posters, very colorful. Did a big publication on the P53. Remember this big network? Look at that title. I can't believe I got published with that long of a title. Some of our early stuff was just saying, like, here's where P53 is in certain subtypes. Then, the new one that is currently in submission, MYC networks and CD8 analysis. Very cool. So, I promise that I'm actually doing stuff, right? Those of us that are watching the recording. Yeah, here's everybody that joined us. And as you can tell, we have lab mascots as well. Scout Pomeranian, very good. Oh yeah, I haven't shown this in class yet, so, but it does exist. But yeah, and then we're also doing the bioed research and everything else, but thanks so much for coming.